Hi guys, Miss Francis here. Today I would like to finish up our discussion about enzymes. Enzymes require these helpers called cofactors. They're non-protein. Some of them are inorganic, such as zinc, iron, and copper, and they're going to help the substrate fit into the enzymes. Your vitamins are types of cofactors. Our hemoglobin, not only is it a conjugated protein, but it contains one of these cofactors known as iron. So that's going to help the substrate fit to the enzyme. There are two different types of inhibitors that are going to exert control over enzyme action, because after all, we don't want the chemical reaction to continue going on forever and ever and ever. The first type of inhibitor that we're going to discuss is called a competitive inhibitor. A competitive inhibitor is one that binds to the same site as the substrate. In other words, the competitive inhibitor has a similar shape. It kind of mimics the substrate, binding to the active site, thus preventing the competitive inhibitor from binding. So here you can see the competitive inhibitor binds, so my substrate is not able to bind to the enzyme. An example would be carbon monoxide. So if you look at carbon monoxide, it has a similar shape to oxygen and carbon dioxide. Well, carbon monoxide can bind to hemoglobin, and it prevents oxygen and carbon dioxide from being able to bind to hemoglobin. And that is very detrimental to our health, because we need our oxygen to do cellular respiration to live, and we need carbon dioxide to be released from our bodies, otherwise it becomes toxic. The other type of competitor that we have is called a non-competitive the next type of inhibitor that we have is called a non-competitive inhibitor. And what the non-competitive inhibitor does is bind to what's known as the allosteric site. So the non-competitive inhibitor binds to the allosteric site, which results in a changed active site. And now our substrate can no longer bind to that active site because the shapes don't match. So any enzyme that can do this, that can change their shape, is known as an allosteric enzyme. Here's another illustration of that, where you see a competitive inhibitor interfering with the active site, whereas a non-competitive inhibitor binds to an allosteric site, a site that isn't the active site, but yet it results in the substrate not being able to bind. Now, inhibition is good because it helps explain some negative feedback cycles, and ultimately the end product is going to stop the reaction, and it's a way of maintaining homeostasis. So let me show you what I mean. In this pathway, we have our substrate. This substrate binds to enzyme 1 to make intermediate A, some sort of product. Intermediate A binds to enzyme 2 to make intermediate B, another product. Intermediate B binds to enzyme 3 to make ultimately this end product. Now let us say we have enough of this end product. We don't need to use energy through our enzymes, or to waste energy using our enzymes to make more of this product. Well, the end product is going to bind to an allosteric site in an enzyme, thus changing the active site, and now my substrate can no longer bind to that enzyme, and the end product is no longer going to be produced. So if we're talking about enzymes, we went through some factors that affect enzyme activity: temperature, pH, salt concentration, enzyme concentration, substrate concentration, and now the presence of inhibitors. Now let's combine two of these. What do you think a graph would look like if I were to introduce a non-competitive versus competitive inhibitor? So let's say this is the action of the enzyme without an inhibitor. When I introduce a competitive inhibitor, right? Let's go back and review once again what a competitive inhibitor does. A competitive inhibitor is something that mimics the shape or has a similar shape to the substrate, but my active site remains the same. The shape of the active site doesn't change. So as I add more substrate, wouldn't it be true that the substrate could ultimately outcompete this competitive inhibitor and be able to bind? So when we draw a graph of that, it would look something like this, where in the beginning, yes, the rate of enzyme activity is lower than it would be when I don't have an inhibitor. However, as my substrate increases, so as I move from left to right, as my substrate increases, now if I look at the difference between my enzyme activity without an inhibitor and enzyme activity with a competitive inhibitor, I can realize that by the end of the enzyme's activity, what I end up with isn't that drastically different as what I would produce if I didn't have an inhibitor present at all. However, if I were to have a non-competitive inhibitor, here's my non-competitive inhibitor, it wouldn't matter how much more substrate I add. I'm still going to end up with a slower rate of enzyme activity than I would if I didn't have the inhibitor, because that active site is still a different shape. It doesn't matter how many additional substrates I add. Immobilized enzymes are enzymes that can be attached to a solid, such as a bead or a filter. So for example, let's say you're lactose intolerant and you can't drink milk containing lactose. Well, I can put my milk through this filter essentially that contains the lactase. And now what comes out is lactase-free milk. That's how companies can make Lactaid, for example, which is milk that doesn't have lactose. And the advantages of using these immobilized enzymes is the fact that these beads can be used over and over and over again for many substrates. I could pour many bottles of milk down this cylinder, which ultimately results in the company being able to save money. There are a bunch of review questions at the end of this PowerPoint that I would encourage you to go through. Tomorrow we will start discussing our last group of macromolecules known as nucleic acids.